Hi everyone, my name is Adji Baskoro. I'm a postdoc researcher in plant molecular biology lab at Chung-Ang University. So today I would like to talk about one of my recent projects entitled Transcriptomic and epigenomic analysis reveal that polycomb repressive complex 2 regulates not only developmental but also stress-responsive metabolism in Brassica rapa. As a sessile organism in nature, plants have evolved various intricate strategies to adapt it when live in unfavorable environmental conditions. Besides, plants also need to produce new organs and tissue throughout the course of their life to sustain its existence. Therefore, plants need precise and competent mechanisms of gene regulation in stress management as well as in growth and development. Here, epigenetic plays a significant role in regulating gene expression from growth and development to stress management through on or off states. Epigenetic mechanisms, including DNA methylation, histone modification, and small RNA, play a crucial role in regulating the expression of genes associated with important agronomic traits. These epigenetic regulations have been shown to impact essential processes from vegetative until reproductive development growth, such as seed germination, plant vigor, flowering, fertility, and fruit development. Besides plant developmental growth, epigenetic mechanisms also involved in plant immunity and defense response mechanisms while exposed to various environmental stress. In here, epigenetic regulation has the potential to revolutionize plant breeding and improve crop yields by regulating gene expressions in plants. Particularly, recent studies show that histone modification are one of the important factors in epigenetic modification, which helping plants to cope with harmful environmental stressors. Histone modification is one of the epigenetic regulatory mechanisms that modulate the chromatin structure and affect various DNA template processes, including gene transcription and DNA replication. Genetic studies provided important information and identified the factors which involve in epigenetic process that were called polycomb group. The polycomb group proteins are highly conserved in eukaryotes, including Drosophila, mammals, and plants, and they form large multimeric protein complexes, namely polycomb repressive complex or PRC. Polycomb repressive complexes play pivotal roles in plant developmental programs by epigenetically modifying chromatin, and PRC traditionally divided into PRC1 and PRC2 in eukaryotes. Both of the PRC1 and PRC2 are essential protein complex for plant development. For the PRC1, it catalyzes monoubiquitination on histone 2A, which is important part of the transcription repression machinery. Meanwhile, the PRC2 mediated deposition of methyl group in histone H3 on lysine 27. Unlike PRC1, the components of PRC2 are well conserved in higher eukaryotes, including plants. PRC2 complex has 4 core components which has different unique role. It is consisting of enhancer of cyste, extra sexcom, suppressor 12 of ZST, and nerve 55. The enhancer of ZST protein containing said domain is required to catalyze the deposition of methyl group in histone H3K on lysine 27. Extra sexcom required to binding to H3K27 trimethylation histone marks, suppressor of 12-ZST, which has a methyl transferase activity directed towards lysine residue of histone 3, and nerve 55 are required for nucleosome association of PRC2 complex to target chromatin. In the Arabidopsis genome, enhancer of ZST has 3 homologs of curly leaf, swinger, and media. 3 homologs of suppressor of ZST, which is vernalization 2, embryonic flowering 2, and fertilization independent seed. 1 homolog for extra sexcom, represented by fertilization independent endosperm 1 or phi 1, and homologs for nerve 55 is the multi-subunit suppressor of IRE or MSI 1 until 5. Genetic and molecular evidence has been suggesting that PRC2 proteins form at least 3 PRC2-like complexes: the embryonic flower EMF, vernalization VRN, and fertilization independent seed or VIS or FIS. Interestingly, although these complexes have clear distinct function, but they still do share some target genes. FIS has a specific role in reproduction, particularly in female gametophyte, endosperm, and seed development. EMF control the flower homeotic genes such as agamose, apetala3, and pistillata during the vegetative and development. Meanwhile, the VRN regulates flowering times and the plant's response to vernalization by repressing flowering locus C. Previous study was reported that Arabidopsis CLF may play major role in developmental programs because the null mutant of CLF displays severe defects during developmental growth compared with the wild-type plants, such as cord leaf and early bolting. Consistently, similar phenotype was also confirmed that loss of CLF function in Chinese cabbage, resulting to early bolting with cord leaf compared to the wild type. Although the phenotypic of CLF was confirmed in the Brassica rapa, but the functional importance of this gene was not fully revealed. Therefore, further investigation was performed to identify the functional importance of BRCLF. First, identification of BRCLF homologs. This is required to identify how many copy numbers of CLF exist in Brassica rapa. Blasts of Arabidopsis CLF protein to Brassica rapa database were performed and show that only single copy of CLF exists in Brassica rapa. As a material, a mutant of CLF in Brassica rapa subspecies 3 locularis was obtained by EMS mutagenized T-link mutant population. The point mutation in the mutant plants was identified by Sanger sequencing. The sequencing validates the point mutation in BRCLF mutant, specifically in the exon number 11 at 3423 base pair from the start coding sequence of BRCLF. The amino acid analysis was detecting the point mutation in the residue at 615 when cytosine change to thymine produce a premature stop codon. This mutation led to the loss of 292 amino acids in the C-terminal regions, which has a catalytic set domain. Phenotypic appearances of PRCLF mutant lines were also confirmed. For example, the PRCLF mutants show abnormal phenotypes such as upward curve and smaller leaf than the R18 wild-type plant. The hypocotyl length, leaf length, leaf width, and rosette diameter of PRCLF mutants were significantly shorter than R18 in the 4-week-old vegetative stage plants. Consistently, at the reproductive stage, the PRCLF plant height was quite shorter than the wild-type plants. The petal and bud lengths of PRCLF mutants also smaller than R18. Meanwhile, the pistil of the PRCLF was extraordinarily elongated, which might substantially reduce the chance for pollination and successive fertilization between egg and sperm cells. Further, the seed fertility was significantly reduced in PRCLF mutant than the wild-type plant. It is reflected to the number seed of PRCLF mutant, which significantly lower in compared to the wild-type plant R18. This is probably caused by the deformed development of floral organs which happen in PRCLF mutant plants. To reveal the regulatory network of CLF in Brassica rapa, RNA-seq analyses were performed to the PRCLF mutants in compared to the wild-type R18. A total of 2,740 different expression genes, including 1,844 upregulated and 896 downregulated genes in PRCLF mutant compared with the wild-type R18, were detected as significant differential expression in pairwise sample comparisons. Correlation heat map analyses show a distinct clustering between R18 and PRCLF samples, indicating that RNA-seq library were well constructed and sequenced. Because Arabidopsis CLF was reported to regulate thousands of genes related to various developmental processes, examination on how many transcription factors is affected in PRCLF in comparison to R18 were performed. List of transcription factors was obtained from the Brassica genome database and collected total 3,410 transcription factor genes in Brassica rapa genomes. Among those transcription factors, total 280 and 88 transcription factor genes were upregulated and downregulated in PRCLF compared with the R18. Given that the PRCLF containing PRC2 possess a repressive role in target genes transcription. The expression of several key transcription factors involved in seed dormancy germination, leaf development, floral organ identity, and floral transition were examined between the wild-type R18 and PRCLF mutants. Several genes represent to those developmental processes were selected, and their transcript levels were compared between both plants. The results show that all tested in Brassica rapa developmental genes were upregulated in PRCLF compared to the wild-type ROAT. In consistency with RNA-seq results, QRT-PCR analysis also show that transcript level of all tested developmental genes were substantially upregulated in BRCLF mutants. Consistently, chromatin immunoprecipitation sequencing or chip sequencing analysis that was obtained from public data also show that all the developmental genes that was selected are enriched with H3K27 trimethylation in leaf and flower organs. This result indicated that PRCLF and its PRC2 complex play a negative role in the expression of many key developmental transcription factor genes in the Brassica rapa plant. Gene ontology analysis was conducted to further seize the biological role of PRCLF from the list of different expression genes. Gene ontology analysis using the list of upregulated or downregulated genes displayed top 20 and top 9 gene ontology categories. Especially the gene ontology analysis using the list of upregulated genes was exhibit that top rankers in the top 20 categories were enriched with stress-related categories such as hydrogen peroxide catabolic process and the reactive oxygen species metabolic process. The results of gene ontology analysis using differential expressed genes suggested that PRCLF might play a regulatory role on the stress signaling genes involved in the hydrogen peroxide catabolic process and reactive oxygen species metabolic process in Brassica rapa. Because gene ontology analysis suggested a possible role of PRCLF on stress signaling, we selected 13 genes related to hydrogen peroxide catabolic process and reactive oxygen species metabolic process from the Brassica rapa genome. The analysis of RNA-seq data show that the transcript level of these 13 genes were commonly higher in PRCLF sample than those of wild-type R18 samples. The QRT-PCR analysis also confirmed the expression of those selected genes are higher in the BRCLF samples. Further, several stress signaling genes that was previously selected were also checked whether they have H3K27 trimethylation enrichment on their genomic regions or not. Chip sequencing analysis showed that 10 genes were densely enriched with H3K27 trimethylation. Because stress signaling genes play a crucial role in the early stage of defense system, it is reasoned that PRCLF containing PRC2 complex might contribute to suppress and maintain basal level expression of stress signal genes when plants in normal condition. In plants, oxidative stress, hydrogen peroxide metabolism, and reactive oxygen species or ROS response are initial signaling events upon exposure to diversity of stresses. The stress signaling pathways play key roles as a stress response via linking the sensing mechanism and the genetic response. Generally, a stress signal transduction pathway comprises the following key steps. First is the signal perception, second is the signal transduction, third is the stress response. After sensing the external stresses via specific receptors, plants transduce the foreign signal into intracellular downstream signaling pathway to stimulate the accumulation of defensive secondary metabolites such as glucosinolates. Since the gene ontology analysis show that BRCLF is involved in the regulation stress-responsive genes, thus the investigation of BRCLF network were extended to the regulation of gene related to stress-responsive secondary metabolism glucosinolate. So what is glucosinolate? Glucosinolate is the most abundant sulfur-rich secondary metabolite compounds which can be found in Brassica crops. Based on epidemiological studies, it has been suggested that the breakdown product of glucosinolate have a health-promoting function as well as a protective ability against cancer for human. This is for human health. Besides, the breakdown product of glucosinolates also has a benefit for the plant itself as a defense property against pathogen. As a precursor of defensive metabolites, glucosinolates are constitutively biosynthesized in almost all tissues, including leaf, root, and also reproductive tissue. However, most of the glucosinolates are majorly synthesized in the leaf part and transported to the zinc organs such as root. During the stress condition, glucosinolates are broken down by myrosinase into several forms such as isothiocyanates, which has biological activity against biotic stressors. The biosynthesis of glucosinolate mostly divided by three major steps. First is chain elongation, core structure formation, and secondary modification. Meanwhile, only indolic glucosinolates, which require two steps, which is core structure formation and secondary modification. Both of the allopathic and indolic biosynthesis were controlled by MIP transcription factor, such as for the allopathic is MIP28, MIP29, and MIP76. For the indolic glucosinolate, it's controlled by the MIP34, MIP51, and MIP122. When the plant is under stress condition, the final form of glucosinolate will break down to obtain a functional defense compound, and it has a biological activity against biotic stressor. Glucosinolate biosynthetic genes were reported to be positively modulated by a group of MIP transcription factors like MIP28 and MIP29 for glucosinolates, MIP34, MIP51, and MIP122 for the indolic glucosinolates. RNA sequencing analysis show that the transcript level of these MIP domain transcription factor genes, BR-MIP29 for glucosinolates and BR-MIP34 and BR-MIP122 for indolic glucosinolate, were substantially upregulated in BRCLF mutant in comparison to the wild-type R18. Chromatin immunoprecipitation sequencing or CHIP sequencing analysis were confirmed that 14 transcription factors were enriched with H3K27 trimethylation in either leaf or flower organs. This result suggests that BRCLF play a negative role in the expression of BR-MIP transcription factor genes regulating aliphatic and indolic glucosinolate biosynthesis in the Brassica rapa plant. Consistent with the transcription factors, the expression of genes that involve in the biosynthesis of aliphatic and indolic are substantially upregulated in the PRCLF mutants in comparison to the wild-type R18. The presence of H3K27 trimethylation was also investigated in the 59 aliphatic and 30 indolic glucosinolate biosynthetic genes. Out of the total 59 aliphatic pathway genes, 32 genes were enriched with H3K27 trimethylation in their genomic regions in either leaf or flower tissue. In case of 30 indolic glucosinolate genes, 80 genes were accumulated with H3K27 trimethylation in their genomic region in either leaf or flower tissue. To confirm that PRCLF is involved in the glucosinolate metabolism, analyses of the glucosinolate profiles were performed by the HPLC to investigate the difference between PRCLF mutant and the wild-type R18. However, there is no difference in terms of the glucosinolate. Both of PRCLF mutant and wild-type R18 are contain the same 4 aliphatic glucosinolate compound and 4 indolic glucosinolate compounds. Although both of PRCLF mutant and wild-type R18 share similar glucosinolate profiles, but the PRCLF mutant had significantly increased amounts of both aliphatic and indolic glucosinolates compared to the levels of R18. PCA analysis display that glucosinolate compound of R18 and PRCLF were clustered in different groups but closely associated each other. Further, the biplot picture, which generated from the PCA data, pointed out that loss of PRCLF affected the glucosinolate metabolism. This data indicated that PRCLF and its PRC2 complex suppress genes involved in the stress signaling cascade and downstream stress-responsive glucosinolate metabolism in Brassica rapa plants. To validate the enrichment of H3K27 trimethylation in the developmental gene related to seed dormancy, leaf size, and floral organ identity, chip assay was performed to wild-type R18 and PRCLF mutant using the H3K27 trimethylation antibody. All the 3 tested genes exhibited that enrichment of H3K27 trimethylation was evidently compromised in the R18 compared to the PRCLF mutant. This indicated that loss of CLF resulted in failure to deposition of H3K27 trimethylation to the target genes. To quantify relative enrichment of H3K27 trimethylation in the glucosinolate transcription factor, cheap qPCR analysis was also applied for the aliphatic glucosinolate transcription factors, which includes 3 transcription factors involved in aliphatic glucosinolates: PRMYP28, 1 PRMYP28-2, PRMYP28-3, and 1 for the PRMYP29. The results show that PRCLF mutant contain low level of H3K27 trimethylation compared to the levels of R18. Similarly, 5 transcription factors which control the indolic glucosinolates such as BR-MIP51, BR-MIP122, BR-OBP21, BR-OBP22, and BR-OBP23 also show that PRCLF contain low level of H3K27 trimethylation compared to the levels of R18. Thus, it is confirmed that PRCLF, a component of Brassica rapa PRC2 complex, is required for H3K27 trimethylation enrichment for the suppression of target genes. Overall, we can see that Brassica rapa CLF was determined as an important factor for the transcriptional regulation of genes related to plant development, stress signaling, and stress-responsive genes glucose storage. This epigenetic mechanism might commonly suppress stress-responsive gene in the absence of challenging stress to benefit plant developmental growth and simultaneously save unnecessary costs on defense programs. So I think that is all from my presentation today. I hope you guys can enjoy it. If you have any questions, don't be hesitate to write on the comment sections, and I will answer your question as fast as I can. Thank you for your attention. See you and have a nice day.